So we learned that the membrane surrounding each cell or organelle is a very busy place. Like a border between two countries, raw materials enter and wastes exit in a continuous flow of traffic. You might ask, how do membranes regulate this activity? In this lesson, we will explore how plasma membranes control transport into and out of cells, specifically the passive transport. So by the end of this video, you can list the types of passive transport that can be used by cells and explain osmosis and the effect it has on cells in environments of different tonicities. So we begin by noting that biological membranes play 5 related yet distinct roles. First, they define the boundaries of the cell and its organelles and act as permeability barriers. Second, they serve as sites for specific biochemical functions. Third, membranes also possess transport proteins that regulate the movement of substances into and out of the cell and its organelles. Fourth, membranes contain the protein molecules that act as receptors to detect extracellular signals. And fifth, they provide mechanisms for cell-to-cell contact, adhesion, and communication. Basically, substances enter a cell in one of 3 ways: through passive, active, or bulk transport. Although there are different types of passive transport, in all of them, substances move from an area of higher concentration to an area of lower concentration, and no energy is required. Active transport moves substances against a concentration gradient, meaning from low concentration to high concentration, and requires both a transport protein and lots of ATP. Lastly, bulk transport requires energy, but movement of the large substances involved is independent of concentration gradients. For this part in our discussion, we will talk about passive transport in detail. The most direct forms of membrane transport are passive. Substances tend to move towards the region where they are few, since their movement is downhill or along their concentration gradient. The process does not require metabolic energy. So all forms of passive transport involve diffusion. During diffusion, molecules move down their concentration gradient until equilibrium is achieved and they are distributed equally. For a familiar example, picture what happens when you first place a tea bag in a cup of water. Near the tea bag, there are many more brown tea molecules than elsewhere in the cup. Over time, however, the brownish color spreads to create a uniform brew. Now, diffusion also occurs across membranes. Each of the large arrows in this diagram shows the net diffusion of dye molecules, and the diffusion of one solute, as you can see here. The membrane has pores large enough for molecules of dye to pass through. The dye diffuses from where it is concentrated to where it is less concentrated. When it has reached equilibrium, it does not mean that the molecules have stopped moving. Instead, the solute molecules continue to cross the membrane at roughly equal rates in both directions. Now let's study the next diagram. Solutions of 2 different dyes are separated by a membrane. This membrane is permeable to both types of dye molecules. Each dye diffuses down its own concentration gradient. The yellow begins moving from left to right, and the purple dye from right to left. After some time, there will be a net diffusion of purple dye towards the left portion, even though the total solute concentration was initially greater on the left side. Thus, this tells us that each substance diffuses down its own concentration gradient unaffected by the concentration gradients of other substances. So recall that lipids and small nonpolar molecules such as oxygen and carbon dioxide, for example, diffuse easily across the hydrophobic portion of a biological membrane. So far, we have seen the diffusion of solutes across a semi-permeable membrane, but how about the diffusion of water? As we will see next, the movement of water across the plasma membrane has important consequences for cells. In here, we have a U-shaped glass tube with a selectively permeable membrane separating two sugar solutions. Pores in this membrane are too small for large sugar molecules to pass through, but are actually large enough for water molecules to pass through. In time, water molecules tend to cling around the hydrophilic portions of the sugar molecules. This makes some of the water unavailable to cross the membrane. As a result, the solution with a higher concentration of solutes has a lower free water concentration. So the diffusion seen in this example is called osmosis. So osmosis, if you compare it with diffusion a while back, is the diffusion of free water across a selectively permeable membrane. So let's compare the two. In a simple diffusion, we are dealing with the movement of solute molecules from high concentration to low concentration with or without a membrane. Meanwhile, in osmosis, we are looking at how solvent, in this case water, moves across a membrane. Since it is the water and not the solutes that can pass through due to its smaller size, water will diffuse down its own concentration gradient toward the side with high solute concentration. That is why this is the result of osmosis. Because there is greater free water concentration on the left portion that will travel to the right portion. The movement of water across cell membranes and the balance of water between cell and its environment are crucial to organisms. To explain the behavior of cell in a solution, we must consider tonicity. So tonicity, the terms hypo, iso, and hyper pertains to the environment surrounding the cell. Let's now apply what we've learned about osmosis to living cells. If a cell without a cell wall, such as an animal cell, is immersed in an environment that is isotonic to the cell, iso means the same, there will be no net movement of water across the plasma membrane. Water diffuses across the membrane but at the same rate in both directions. In an isotonic environment, the volume of an animal cell, as you can see here, is stable. Now let's transfer the cell to a solution that is hypertonic to the cell. This means that there is a greater solute concentration outside the cell. Water inside the cell will diffuse towards high concentration of solutes outside, causing the cell to lose water, shrivel, and probably die. Therefore, an increase in the salinity of a lake can kill the animals there, and if the lake water becomes hypertonic to the animal cells, they might shrivel and die. However, taking up too much water can just be as hazardous as losing water for the cell. So if we place a cell in a solution that is hypotonic to the cell, meaning solutes are lesser outside the cell and higher inside, water will enter the cell faster than it leaves, and the cell will swell and lyse or burst, just like an overfilled water balloon. Next, we look at how cells with cell walls react to different tonicities. Organisms such as plants, prokaryotes, fungi, and some unicellular eukaryotes have cells that are lined with cell walls. So let's take plant cells for example. When these cells are immersed in a hypotonic solution, the cell wall helps maintain the cell's water balance. The plant cell swells as water enters by osmosis, just like that of an animal cell. However, because of the presence of the cell wall, which animal cells do not have, the cell will expand only so much before it exerts back a pressure on the cell. This pressure is called turgor pressure, and this opposes further water uptake. At this point, the cell is very much turgid or very firm. This is the healthiest state for most plant cells. Now, if a plant cells and surroundings are isotonic, okay, there is no net tendency for water to enter and the cells become flaccid or limp. Hence, the plant wilts. Now, if the cell is immersed in a hypertonic environment, the plant cell will lose water to its surroundings and shrink. As the plant cell shrivels, its plasma membrane pulls away from the cell wall, as you can see here, at multiple places. This phenomenon is called plasmolysis, which causes the plant to wilt and can lead to plant death. So what is the best tonicity for animal and the plant cell? Animal cells in an isotonic environment are on its most stable state, okay? While plant cells are on its healthiest state when in a hypotonic environment, because it is at this point that their cells are turgid. Plants that are not woody, such as most houseplants, depend for mechanical support on cells that are kept turgid by a surrounding hypotonic solution. We now come to the last type of passive transport, which is the facilitated diffusion. Ions and polar molecules cannot freely cross the hydrophobic layer of a membrane. Instead, transport proteins form channels that help these solutes cross. So facilitated diffusion occurs when polar molecules and ions diffuses across a membrane with assistance from a channel or a protein. Channel proteins simply provide corridors that allow specific molecules or ions to cross the membrane. The hydrophilic passages provided by these proteins can allow water molecules or small ions to diffuse very quickly from one side of the membrane to the other. Water channel proteins are called aquaporins, and they facilitate the massive levels of diffusion of water in plant and animal cells. Meanwhile, channel proteins that transport ions are called ion channels and are highly specific. Carrier proteins undergo a subtle change in shape that somehow translocates the solute binding site across the membrane. Such a change in shape may be triggered by the binding and release of the transported molecule. Like ion channels, carrier proteins involved in facilitated diffusion result in the net movement of a substance down its concentration gradient. Both channel proteins and carrier proteins are very specific to the molecule they assist across the membrane. Even though facilitated diffusion involves transport proteins, it is still passive transport because the solute is moving down their concentration gradient, meaning from high to low concentration. In summary, all forms of passive transport involve diffusion, the dissipation of a chemical gradient by random molecular motion. And in simple diffusion, a substance passes through a membrane along its concentration gradient without the aid of a transport protein. Osmosis is the simple diffusion of water across a selectively permeable membrane. Terms describing tonicity such as isotonic, hypotonic, and hypertonic predict whether cells will swell or shrink when the surroundings change. And lastly, in facilitated diffusion, a membrane protein admits a substance along its concentration gradient without expending energy.